The lymphatic glands. As it is impossible experimentally to prevent the lymphatic glands as a whole from contributing to the formation of the blood, we are dependent almost entirely on clinical and anatomical researches for an elucidation of their function. Since Virchow's definition of the lymphocyte, it has been admitted that the lymphocytes of the blood, both the small and the larger kinds, are identical with those of the lymphatic glands and the rest of the lymphatic system. This is proved by the complete agreement in general morphological character, in staining properties of the protoplasm and nucleus, and from the absence of granulation. Abundant clinical experience testifies that the lymphocytes of the blood really do arise from the lymphatic system. Ehrlich had previously observed that when extensive portions of the lymphatic glandular system are put out of action by new growths and similar causes, the number of the lymphocytes may be considerably diminished. These observations have since that time been confirmed by various authors. For example, Reinbach describes several cases of malignant tumour, particularly sarcoma, in which the percentage of lymphocytes, which normally amounts to about 25%, was very considerably lowered. In one case of lymphosarcoma of the neck, they only made up 0.6% of the total number. These conditions are quite easily and naturally explained by the exclusion of the lymphatic glands. It is difficult for the advocates of the view that the lymphocytes are the early stages of all white blood corpuscles to reconcile it with these facts. According to their scheme, the low number of lymphocytes is to be explained in such cases by their unusually rapid transformation to the polynuclear elements, the old forms, or, to appropriate the expression of Uskov, by a too rapid aging of the lymphocytes. Further evidence for the origin of the lymphocytes of the blood from the lymphatic glands is to be obtained from those cases in which we find an increase of the lymphocytes in the blood. These lymphocytoses occur, in comparison with other leukocytoses, relatively seldom. Under certain conditions in which a hyperplasia of the lymphatic glandular apparatus makes its appearance, we often see at first an increase of the lymphocytes in the blood. Ehrlich and Karewski, in some unpublished work, have investigated together a large number of typical cases of lymphoma malignum, and were able constantly to observe a lymphocytosis, which in some cases was of high degree, and bore almost a leukemic character. Relying on these facts, Ehrlich and Wassermann made the diagnosis in vivo of malignant lymphoma in a rare skin disease, chiefly from the absolute increase of the lymphocytes alone, although no swelling of the glands was palpable. The post-mortem showed that the chief condition was a swelling of the retroperitoneal lymph glands to lumps as large as a fist. The lymphocytosis following extirpation of the spleen also belongs to this category, since a vicarious enlargement of the lymph glands is always to be observed in these cases. On investigating the conditions under which in healthy individuals an increased number of lymphocytes enter the bloodstream, we have in the first place to notice the digestive canal, whose wall contains a thick layer of lymphatic tissue. According to the results of Ryder, the proportion of the lymphocytes to polynuclears is practically normal in the leukocytosis of digestion. Indeed, the lymphocytes are rather in excess. The eosinophils, on the other hand, show a marked relative reduction in this condition. The leukocytosis of digestion consequently differs essentially from the other kinds in which the neutrophil elements are chiefly increased. The simultaneous increase of lymphocytes and polynuclears is doubtless brought about by a superposition of a raised income of lymphocytes and an ordinary leukocytosis caused by the assimilated products of metabolism. The influence of the digestive tract is still more evident in certain diseases, more particularly in intestinal diseases of infants. A considerable increase of the lymphocytes in the bloodstream is here to be observed. Thus, Weiss found an important increase of the white blood corpuscles in simple catarrh of the stomach and intestines, which presented the main features of a lymphocytosis. Whooping cough, according to the recent observations of Meunier, also belongs to the small number of diseases which are accompanied by a pronounced lymphemia. In the convulsive period of this disease, both the polynuclear cells and the lymphocytes are increased, the latter in preponderating amount. The former cells are increased to twice, the lymph cells to 4 times their normal amount. Doubtless in these cases also the lymphocytosis is due to the stimulation and swelling of the tracheobronchial glands. An increase of the lymphocytes from chemical stimuli is exceedingly rare, though, as is well known, a large number of substances, bacterial products, proteins, nucleins, organic extracts, and so forth, can call forth a polynuclear leukocytosis. In quite isolated cases, an increase of the lymphocytes in the blood in consequence of the injection of tuberculin into tuberculous individuals has been seen. From the rarity of these cases, it can scarcely be doubted that here a tuberculous disease of the glands also plays a part, so that the increased immigration of lymphocytes is brought about not by a chemical property of the tuberculin, but by the extensive specific reaction of the diseased glands. Only one single substance has so far been mentioned in the literature as capable in itself of producing a lymphocytosis. Waldstein asserts that he has produced by injection of pilocarpine a lymphemia which undergoes a progressive increase with a rise in number of the injections. The origin also of lymphocytosis is therefore sharply marked off from that of the ordinary leukocytosis, which consists in an increase of the neutrophil elements. Whilst the latter is admittedly the expression of chemiotactic action, and arises by action at a distance of soluble substances on the bone marrow, lymphocytosis is due to a local stimulation of certain glandular areas. Thus, in the leukocytosis of digestion, of intestinal diseases of children, we refer it to the excitation of the lymphatic apparatus of the intestine. In tuberculin lymphemia, we recognize mainly a reaction of the diseased lymph glands. Hence, we conclude that a lymphocytosis appears when a raised lymph circulation occurs in a more or less extended area of lymphatic glands, and when, in consequence of the increased flow, more elements are mechanically washed out of the lymph glands. The pilocarpine lymphocytosis does not contradict this view, for pilocarpine causes extraordinary, though transient, variations in the distribution of water, whereby the inflow into the blood of fluid containing lymph cells is increased. We therefore regard lymphocytosis as the result of a mechanical process, whilst leukocytosis is the expression of an active chemiotactic reaction of the polynuclear elements. This view finds its best support in the fact that the polynuclear leukocytes possess lively amoeboid movement, which is completely wanting in the lymphocytes. Corresponding to the absence of contractility in the lymphocytes, it is also observed that in inflammatory processes, in contradistinction to the polynuclear neutro and oxyphils, the lymphocytes are not able to pass through the vessel wall. A very interesting experiment on this point was described by Neumann years ago. Neumann produced suppuration in a patient with lymphatic leukemia, in whom the blood contained only a very small number of polynuclear cells. Investigation of the pus showed that it consisted exclusively of polynuclear cells, and that not a single lymphocyte had come into the exudation, although this kind of cell was present so abundantly in the blood. Histological examination of all fresh inflammatory processes, in which mainly polynuclear elements are found, leads to accordant results. It is well known that small-celled infiltration occurs in the later stage of inflammation, apparently consisting of lymph cells. Nevertheless, this does not in the least prove that these lymphocytes have emigrated here from the blood vessels. This is not the place to enter into the very extensive controversy on this point. We are content to refer to the most recent, very thorough paper of Ribbert. Ribbert regards these foci of small-celled infiltration as the analogues of the lymphatic nodules, and explains their origin by an increase in size of the foci of lymphatic tissue, normally present though in a condition but little developed. It consequently follows from clinical and morphological researches, as well as from the observations on inflammatory processes, that the lymphocytes are in no way connected with the polynuclear leukocytes. We shall reach the same result in another way in the following section. Gamma, the bone marrow. The spleen and lymphatic glands were at first regarded as the sole places of formation of the blood corpuscles. The almost simultaneous researches of Newman and Bizzozero first attracted general attention to the importance of the bone marrow. These authors showed that the early stages of the red blood corpuscles are produced there, a discovery which was quickly and generally recognised, and which soon became pathologically useful through the observations of Cohnheim and others. In this connection the observation was of great value that after severe loss of blood, the fatty marrow of the larger hollow bones again changes to red marrow, as it is evidence of the increased demands on the regenerative function of the bone marrow. We are unaware of a second place of formation of the red blood corpuscles in man. In other mammalia, however, as we have above mentioned, the spleen may also take a small share in the production of erythrocytes. The type which the normal blood formation follows in adults, and the deviations therefrom shown in pernicious anemia, have been described in the chapter on the red blood corpuscles. Ehrlich's view that the blood formation in pernicious anemia belongs to a different type, which is analogous to the embryonic, was also described there. In this section we have therefore to deal chiefly with the white blood corpuscles and their connection with the bone marrow. In man, as in a large number of animals, for example the monkey, guinea pig, rabbit, pigeon, and so forth, the bone marrow exhibits the peculiarity that the cells it produces bear a specific granulation, in sharp contrast to the lymphatic glandular system, which contains elements free from granules, in the whole animal series. The granulated cells of the bone marrow fall into two groups. The first group of the cells with special granules is very important, since it constitutes a characteristic for certain species of animals. According to the class of animal, they show different tinctorial and morphological properties. Man and monkey, for example, have neutrophil granulation. Guinea pig and rabbit, the pseudo-eosinophil granulation described by Kurloff. In birds, we find two specific granulations present side by side, which both are oxyphil, and of which one is embedded in the protoplasm in crystalline form, the other in the form of granules. The kinds of special granulations so far investigated have the common property that they stain in acid and neutral dyes respectively. They show a much smaller affinity for the basic dyes. The fact that they greatly exceed the other elements of the bone marrow in all classes of animals is evidence of the importance of these granules. The second group of bone marrow cells contains granules which we find in the whole vertebrate series from the frog to man, and which therefore are not characteristic for any one species of animal. They are 1. The eosinophil cells, 2. The basophil mast cells. The bone marrow forms which are free from granules consist mostly of mononuclear cells of different type. They are not nearly so numerous or so important as the granulated kind, more especially as the first and predominant group. Amongst the granule-free forms, the giant cells deserve special mention, for they are an almost constant constituent of the bone marrow of the mammalian class. According to the recent researches of Pugliese, the giant cells are considerably increased after extirpation of the spleen in the hedgehog, an organ of quite extraordinary size in this animal, and doubtless therefore possessing important haematopoietic functions. Pugliese asserts that in the hedgehog after splenectomy the nucleated giant cells pass into leukocytes by amitotic nuclear division. Unfortunately, in his preliminary communication there are no notes of the granules of the bone marrow cells. On examining a stained dry preparation of the bone marrow of the guinea pig, rabbit, man, etc., it is seen that the characteristic finely granular cells are present in all stages of development, from the mononuclear through the transitional to the polynuclear (polymorphously nucleated) forms, which we meet with in the circulating blood. A glance at a preparation of this kind shows that the bone marrow is clearly the factory where typical polynuclear cells are continuously formed from the granule-containing mononuclears. Here also the same process of ripening can be seen in the polynuclear eosinophil leukocytes. Ehrlich has been able, by special differential staining, to bring forward proof that the constitution of the granulation changes during the metamorphosis of the mononuclear to the polynuclear cells. In the young granules there is prominent a basophil portion that becomes less and less marked as the cell grows older. The pseudo-eosinophil granules of the mononuclear cells, of the guinea pig for example, stain bluish red in eosin methylene blue after long fixing in superheated steam. In the transitional stages this admixture is gradually lost, and finally completely vanishes in the granules of the polynuclear leukocytes, which stain pure red. Analogous observations may be made in the eosinophil cells of man and animals, and in the neutrophils of man. Hence, it is even possible to decide whether an isolated granule belonged to an old or to a young cell. It is still impossible to judge with certainty the rate at which the ripening of the mononuclear to the polynuclear cells proceeds, or further to decide if the ripening of the granules always runs parallel in point of time with that of the whole cell. On the grounds of our observations, we would suppose that in general the two processes run their course side by side, but that in special cases the morphological ripening of the cell may proceed more rapidly than that of the granules. It is particularly easy to observe this point in eosinophil cells. Ehrlich had already mentioned in his first paper, 1878, that side by side with the typical eosinophil granules, isolated granules are often found which show a deviation in tinctorial properties. For instance, they stain more of a black colour in eosin aurantia nigrosin, in eosin methylene blue, bluish red to pure blue. Ehrlich had already described these as young elements in his first paper. The same differences are found more sharply marked in leukemia, even in the circulating blood, in the neutrophil as well as in the eosinophil group. Ehrlich has repeatedly found in leukemic blood polynuclear eosinophil cells whose granules must almost exclusively be regarded as young forms. Footnote: many authors, e.g. Arnold, explain this double staining of the eosinophils by the presence of eosinophil and mast cell granulation side by side. That this is certainly not the case is shown by the fact that the basophil granulation of the eosinophil cells does not, in metachromatic staining, show the metachromasia characteristic for the mast cells. End footnote. Ehrlich regarded these as typical examples of a relative acceleration of the morphological ripening of the cells, as compared with the development of the granules. In normal blood we find only the ripe forms of the specific granulated cells of the bone marrow. The mononuclear and transitional forms of the neutrophil group do not, under normal circumstances, pass over into the bloodstream. Ehrlich regarded the mononuclear neutrophil granulated cells as characteristic for the bone marrow, since they are found exclusively in the bone marrow, never in the spleen or lymph glands, and for this reason named them myelocytes. Footnote: A. Frankel has recently reported histological investigations in which he could demonstrate in one case true myelocytes in inflamed lymph glands. He says, "For some time past I have had systematic examinations carried out by my assistant, Dr. Jaffa, on the granulations of the leukocytes contained in these glands in a large number of infectious diseases, which are accompanied by acute swelling of the lymphatic glands, such as scarlet fever, diphtheria, typhoid. They were performed in the following way. Dry coverslip preparations were made from the juice of the glands removed shortly after death, and were stained in the usual way by Ehrlich's triacid mixture. Amongst a large number of cases thus examined, it was possible in only one case of scarlet fever, but in this, beyond all doubt, to demonstrate the presence of mononuclear cells with neutrophil granulation. The extreme rarity of this condition supports our opinion that the formation of neutrophil mononuclear elements cannot be regarded as a normal function of the lymphatic glands. Polynuclear neutrophil cells are nearly always naturally present in inflamed lymph glands, as a product of the inflammation which has immigrated there. Every pus preparation shows that the polynuclear neutrophil leukocytes can change in the tissues to mononuclear, and the isolated observations of Jaffa should be explained in this manner. End footnote. When myelocytes, no matter of what size, appear in considerable numbers in the blood of an adult, a leukemia of myelogenic nature is nearly always present. Note For the very rare exceptions to this rule, which, it may be added, can never be confused with leukemia, see later pages. End note. Exactly similar conditions hold good for the eosinophil cells, inasmuch as the singly nucleated cells, which one may call eosinophil myelocytes, occur almost exclusively in leukemic blood. These forms, which were first recognised by H.F. Müller, are, however, of less importance, for in myelogenic leukemia the chief part of the foreign admixture of the blood is made up of Ehrlich's myelocytes. Very important conclusions on the interesting question of leukocytosis can be drawn from these observations. Bearing in mind that polynuclear neutrophil cells are developed and stored up only in the bone marrow, that in ordinary leukocytosis only the polynuclear forms are increased in the bloodstream, it is evident that leukocytosis is purely a function of the bone marrow, as Ehrlich has always insisted with all distinctness. It is only on this assumption that the frequently sudden appearance of leukocytosis, as has so often been observed in morbid and experimental conditions, can be satisfactorily explained. In these cases the space of time, amounting often only to minutes, is far too short for a new formation of leukocytes to be conceivable. There must be places in which these cells are already completely formed, and able thence to emigrate on any suitable stimulus. This place is single, and is the bone marrow alone. Here all mononuclear forms gradually ripen to the polynuclear contractile cells, which obey each chemiotactic stimulus by emigration, and which thus bring about sudden leukocytosis. The bone marrow thus fulfills, amongst others, the extremely important function of a protective organ, by which definite injurious influences which affect the organism may be quickly and energetically combated. Just as in a fire station ample means of assistance is continuously in readiness immediately to answer an alarm from any quarter. We wish to insist once more that the large mononuclear leukocytes and the transitional forms of the normal blood are not concerned in the increase in ordinary leukocytosis. In leukocytosis of high degree their relative number may indeed be lowered, in consequence of the exclusive increase of the polynuclear cells. It appears then that these elements do not react to chemiotactic stimuli, and that possibly they reach the blood by entirely different ways than the polynuclears do. We believe that these non-granulated mononuclear cells of man are to be regarded as analogous to those of the guinea pig, described by Kurloff. The mononuclear cells of man, however, are finally transformed into the neutrophil granulated cells, whilst the cells of Kurloff remain free from granules in the course of their metamorphosis. In acute leukocytosis in the guinea pig, only the pseudo-eosinophil polynuclear cells are increased, which wander as such out of the bone marrow, but not the polynucleated non-granulated forms, which but slowly grow to maturity in the blood. Thus the peculiarities of guinea pig's blood, in which two kinds of polynuclear cells are recognizable, throw light upon the corresponding conditions in human blood. The distinction in the latter is more difficult, since it is not evident in this case that the fully formed polynuclear neutrophil leukocytes have a twofold origin, for the majority wander fully formed from the bone marrow into the blood, and only a considerably smaller number grow to maturity within the bloodstream from the mononuclear and transitional forms. No definite statement can as yet be made as to the places of formation of the non-granulated large mononuclear leukocytes. Kurloff has demonstrated that in the guinea pig these cells are present both in the bone marrow and in the spleen, but that after extirpation of the spleen the absolute number does not change. The bone marrow then in the guinea pig can also preserve the balance of the large mononuclear non-granular cells in the blood. The numbers we found in our blood investigations in man after splenectomy were also normal. We may then doubtless assume that the large mononuclear granule-less cells of human blood also arise for the most part from the bone marrow. In this tissue they are to be picked out in the medley of the different kinds of cells only with the utmost difficulty, owing to their small number and their but little characteristic properties. Consequently, an exact investigation of their origin could probably only be successful if it were possible experimentally to produce a disease in which these forms in particular underwent important increase. This advance is not quite hopeless, since in man at least an absolute increase of the large mononuclear cells is observed in the post-febrile stage of measles. On the grounds merely of microscopical investigations, we conclude that the bone marrow is by far the most important of the blood-forming organs, for its function is the exclusive production of red blood discs, as well as of the chief group of the white corpuscles, the polynuclear neutrophil. The physiological experimental investigation of the functions of the bone marrow offers insurmountable difficulties. An exclusion of the whole bone marrow, or of larger portions only, is an impossible operation. Nor can we ascribe any value to the researches which endeavour to obtain a result by a comparative enumeration of the arterial and venous blood of a bone marrow area. J. P. Ruetzky, working under Uskoff's direction, has recently made counts of this kind in the dog, from the nutrient artery of the tibia and the corresponding vein. He found that the number of white corpuscles of the vein is slightly greater, that on the other hand the absolute number of young corpuscles (Uskoff), i.e. of the lymphocytes, has been considerably diminished, while the number of ripe corpuscles, which for the most part correspond to our polynuclear, is considerably increased. He gives the following table. Total number of cells in arterial blood 15,000, in venous blood 16,400. Young corpuscles in arterial blood 1,950, that's 13%. In venous blood 656, 4%. Ripe corpuscles in arterial blood 840, 5.6%. In venous blood 2,788, 17%. Old corpuscles in arterial blood 12,210, 81%. In venous blood 12,956, 79%. The argument based on figures such as these assumes that the function of the bone marrow is continuous, an assumption which Uskoff indeed seems to make. But if the bone marrow is constantly absorbing the lymphocytes to such an extent, it is quite incomprehensible how the normal condition of the blood can be preserved, bearing in mind the extent of the bone marrow and the rate of the circulation. All evidence, indeed, tends to show that, on the contrary, the bone marrow performs its functions discontinuously, inasmuch as elements continually grow to maturity in the bone marrow, as we have already explained, but they only emigrate at certain times as the result of chemical stimuli. It is obvious a priori from this consideration how inconclusive must be the results of experiments such as these of Ruetzky. Moreover, the investigations of Ruetzky are quite without foundation, inasmuch as the tibia of the dog, upon which this author performed his experiments, contains in all races of dogs, according to the information very kindly given us by Professor Schürtz, no red marrow, but fatty marrow only, which, as is well known, is incapable of the smallest hematopoietic function. Far more important for the elucidation of the function of the bone marrow are clinical observations on cases in which considerable portions of the bone marrow are replaced by tissue of another kind. We may best divide the observations on this point into two groups. 1. Malignant tumours of the bone marrow. 2. The so-called acute leukemia. There are, unfortunately, very few available observations as yet upon the first group. Still rarer are the cases in which, as is necessary, the whole bone marrow has been subjected to an exhaustive examination, which alone affords adequate evidence of the extent of the defect. Amongst the changes of the bone marrow arising from tumours, one may distinguish two groups, according to the nature of the condition of the blood. The first type is exemplified by a case of Northnagel, published in his work on lymphadenia osseum. Here, during life, the blood showed in the main the features of a simple severe anemia, but in addition isolated normoblasts, small marrow cells, and moderate leukocytosis. The autopsy, at which the whole skeletal system was subjected systematically to an exact examination, showed a complete atrophy of the bone marrow, and replacement of the same by the tumour masses. In this case, then, the condition of the blood in vivo is satisfactorily explained by the absence of function of bone marrow. Northnagel conjectured that the formation of the scanty nucleated red blood corpuscles occurred vicariously in the spleen, that of the leukocytes in the lymph glands. In the second series, to which the cases of Israel and Leyden, as well as the recently published one of J. Epstein from Neusser's wards, belong, the blood shows, besides the usual anemic changes, other anomalies which are peculiar partly to pernicious anemia, partly to myelogenic leukemia. In Epstein's case of metastatic carcinoma of the bone marrow, there was found a considerable anemia, with numerous nucleated red blood corpuscles, both of the normo and megaloblastic types. Their nuclei presented the strangest shapes, due not merely to typical nuclear division, but also to nuclear degeneration. The white blood corpuscles were much increased. Their proportion to the red was 1 in 25 to 1 in 40. The increase concerned in the main the large mononuclear forms, which bore for the most part neutrophil granulation, and were therefore to be called myelocytes. In all the specimens only 2 eosinophil cells were found. Footnote: we draw particular attention to the small number of eosinophil cells, since according to Ehrlich's postulates, this absence of eosinophil cells is incompatible with the diagnosis of a leukemia. End footnote. The explanation of a blood picture of this kind, apart from the purely anemic changes, is by no means easy, as Epstein rightly observes. The appearance of myelocytes is most readily explained by a direct stimulation of the remaining bone marrow by the surrounding masses of tumour. In this the mechanical factor is less concerned than the chemical metabolic products of the tumour masses, which at first act on the adjacent tissue in specially strong concentration, and also in a negatively chemiotactic manner on the wandering cells. This view receives support from the careful work of Reinbach on the behaviour of the leukocytes in malignant tumours. Out of 40 cases examined, in only 1 of lymphosarcoma complicated with tuberculosis were myelocytes found in the blood, amounting to about 0.5 to 1% of the white blood corpuscles. The autopsy showed isolated yellowish-white foci of growth in the bone marrow, reaching the size of a sixpenny piece. Bearing in mind that in none of the other 39 cases were myelocytes demonstrated, one does not hesitate to explain their presence in the blood in this single case by the metastases in the bone marrow. The small extent of the latter is likewise the cause of the small percentage of the myelocytes. In explaining the presence of the megaloblasts in the blood of Epstein's patient, we must keep before us what we have said elsewhere on this kind of cell. They are not present in the normal bone marrow. They arise, on the contrary, according to our view, when a specific morbid agent acts upon the bone marrow, as we must assume is the case in the pernicious forms of anemia. In the cases of anemia from tumours, in which we find megaloblasts in large numbers in the blood, we must likewise assume that chemical stimuli proceed from the tumours, leading to the formation of megaloblasts in the bone marrow. The presence of megaloblasts in the bone marrow does not itself cause their appearance in the blood, for in pernicious anemia the bone marrow may be filled with megaloblasts, and yet only very scanty examples are to be found in the blood. Whether the emigration of the megaloblasts from the bone marrow into the bloodstream is in general to be referred to chemical stimuli, as it is in the particular case of Epstein's, or to mechanical causes, cannot at present be decided. The bone marrow may be replaced by typical lymphatic tissue, as well as by the substance of malignant tumours. The former occurs constantly in lymphatic leukemia, according to the well-known results of Neumann, which have since been generally confirmed. In these cases, extensive tracts of bone marrow are replaced, not by masses of malignant growth, but by an indifferent tissue, so to speak, a tissue which is unable to exercise the above described stimulating influence upon the remaining bone marrow. It is owing to this circumstance that we are able to observe, in the cases of lymphatic degeneration of the bone marrow, the phenomena due to its exclusion in their most uncomplicated form. Footnote: in contrast to this lymphatic metamorphosis of the bone marrow, in myelogenous leukemia, a myeloid transformation of the other blood-forming organs, especially of the lymph glands, is found. A transformation sufficiently characterized as myeloid by the presence of myelocytes, eosinophils, and nucleated red blood corpuscles. End footnote. The most convincing results are obtained from cases of acute lymphatic leukemia, the pretty frequent occurrence of which was first noticed by Epstein, and which has lately been very thoroughly studied by A. Frankel. For the purpose in question, acute leukemia is specially suited, since the abnormal growth of the lymphatic tissue takes place very rapidly, and for this reason brings about a quick and uncomplicated exclusion of the bone marrow tissue, as it were experimentally. Under its influence, the neutrophil elements of the bone marrow vanish rapidly, and in many cases so completely that it needs some trouble to find a single myelocyte, as for example in a case of Ehrlich's. The polynuclear leukocytes are produced in the bone marrow. Consequently, where the bone marrow is destroyed, as in this case, it is clear that their numbers must be absolutely very much diminished in the blood. Dock has also arrived at similar results, as we see from a preliminary report, and he similarly explains the absence of neutrophil cells in lymphatic leukemia by the replacement of the myeloid by lymphatic tissue. Thus, lymphatic leukemia affords a striking proof that the lymphocytes are cells of a peculiar kind, and which are quite independent of the polynuclear cells. It is therefore exceedingly surprising that Frankel, after accurately examining and analysing 8 cases of acute lymphatic leukemia, believes he has found in them imperative reasons for the assumption that the lymphocytes are transformed to polynuclear cells. This can only be explained by the confusion which Uskoff's doctrine of young cells has brought about. We define lymphocytosis as an increase of the lymphocytes of the blood. Frankel, like Uskoff, regards it as the emigration of the young forms of the white blood corpuscles into the blood. He concludes logically from the diminution of the polynuclear cells in this form of disease that the conditions of the transformation of the young forms have undergone a disturbance. But if one assumes that the lymphocytes are young forms and the polynuclears their older stages, it is much nearer to the facts to speak not of a disturbance in lymphatic leukemia, but of an absolute hindrance to the ripening process. It is easy to conceive any particular stimulus or injury bringing about an acceleration of the normal process, that is, a premature old age, but it is equally difficult to represent clearly to oneself conditions which retard or completely prevent the normal aging of the elements. The discovery of such conditions would be really epoch-making, both for general biology and for therapeutics. The only escape from this dilemma would be the assumption of a very premature death of the lymphocytes, for which, however, not the smallest evidence is to be found, even in Frankel's monograph. Frankel distinguishes the acute from the chronic forms of leukemia by the fact that in the former the newly formed elements emigrate from their places of formation into the bloodstream with extraordinary rapidity, hence there is not time for further local metamorphosis. In chronic leukemia the emigration takes place very probably much more slowly. This distinction is contradicted by the facts, for there are chronic forms of lymphatic leukemia whose microscopic picture is identical with that of acute leukemia, and hence the starting point of all Frankel's deductions is rendered insecure. End of section 7.
Histology of the blood. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by Anna Simon. Histology of the Blood by Paul Ehrlich and Adolf Lazarus. Translated by M. Myers. Section 8. Chapter 3. On the demonstration of the cell granules and their significance. During the last 10 years, a large amount of valuable work has been done on the cell granules from histological, biological, and clinical sides. This has particularly assisted hematology, where a number of problems remain whose solution is only possible by the aid of a knowledge of the granules. We must therefore consider the history, methods, and results of this work. Ehrlich was undoubtedly the first to insist on the importance of the cell granules and to obtain practical results in this direction. We are obliged to mention this, since Altmann has, in spite of express corrections, repeatedly asserted the contrary. In 1891, Ehrlich refuted Altmann's claim to priority. Nevertheless, Altmann, in the second edition of his Elementary Organisms, 1894, stated that before him no one had recognized the specific importance of the granules, though some authors had viewed them as "rare and isolated phenomena." We may quote a passage published by Ehrlich in 1878, that is, 10 years before Altmann's papers. "Since the beginning of histology, the word granular has been used to describe the character of cellular forms. This term is not a very happy one, since many circumstances produce a granular appearance of the protoplasm. Modern work has shown that many cells, formerly described as granular, owe this appearance to a reticular protoplasmic framework, and we have no more right to call cells granular in which proteid precipitates occur, either spontaneously as in coagulation, or from reagents, alcohol. The name should be kept exclusively for cells in which during life substances, chemically distinct from normal proteid, are embedded in a granular form. We can readily distinguish but few of these substances, such as fat and pigment. Most of them we can at present characterize but imperfectly, or not at all." "Earlier observations, especially on the mast cells, led me to expect that these granulations, though they had long been inaccessible to chemical analysis, could be distinguished by their behavior with certain stains. I found in fact granules of this kind characterized by their affinity for certain dyes, and which could thereby be easily followed through the animal series and in various organs. I further found that certain granules only occurred in particular cells, for which they were characteristic, as pigment is for pigment cells, and glycogen for cartilage cells, Neumann, and so forth. We can diagnose the variously shaped mast cells only by the staining of their granules in dahlia solution, that is, by a microchemical test. And in the same way we can separate tinctorially other granulated cells, morphologically indistinguishable, into definite subgroups, and for this reason I propose to call these granulations specific. The investigations were performed after Koch's method in the following manner. The fluid, blood, or the parenchyma of the organs (bone marrow, spleen, etc.) was spread on cover slips in as thin a layer as possible, dried at room temperature, and after a convenient length of time stained. I had chosen this apparently coarse method for the special reason that, for the histological recognition of new, possibly definite chemical combinations corresponding to the granulations, all substances must be avoided that might act as solvents, for example water or alcohol, or as oxidizing agents, such as osmic acid. In this instance only such procedures may be employed as will leave the simple drying of each single chemical substance as much as possible unchanged." A more detailed study of the process of staining, and of the relation between chemical constitution and staining power, enabled a further advance to be made. And the first result in this previously unworked direction was the sharp distinction between acid, basic, and neutral dyes, and between the corresponding oxy, baso, and neutrophil granulations. The triacid solution was only found after trial of many hundred combinations, and up to the present day this stain in its original form or in slight modifications has played a prominent part in various provinces of histology. The classification of the cell granules of the blood according to their various chemical affinities, which was drawn up by this method, is accepted today as the most valuable, and the only practical means of grouping the leucocytes. From the first, Ehrlich has insisted that different kinds of cells possess different granules, distinguished not only by their tinctorial properties, but also by their various reactions to solvents. It is in this connection indeed that Altmann's method, consisting of a complicated hardening process and the use of a single always similar stain, constitutes a retrograde step, inasmuch as it tends to obscure the principle of the specificity of each kind of granulation. A further disadvantage of Altmann's hardening method lies in the circumstance that the cell proteids are precipitated by it in a spherical form, and stain in the subsequent treatment. Hence it is extremely difficult to distinguish what is preformed and what is artefact. Since A. Fischer's publication, where the formation of granule-like precipitates under the influence of various reagents is experimentally demonstrated, grave doubts as to the reality of Altmann's forms have been raised from various quarters. Ehrlich's dry process, on the contrary, is entirely free from error. Granules cannot be artificially produced by desiccation, and the stained appearances correspond precisely to what is seen in fresh living blood. The greatest value of the dry method is that the chemical nature of the single granules remains unchanged, so that attempts at differentiation are made on a nearly unaltered object. Altmann's freezing process would be similar to the advance always insisted on by Ehrlich. It offers such great technical difficulties, however, that it has up to now been little used. Another means of studying the nature of the granules depends on the principle of vital staining. The vital methylene blue staining, Ehrlich, that has since become so important, especially in neurology, led to the first attempts at staining the granules in living animals. One of the first publications on this subject is that of O. Schultze, who placed the larvae of frogs in dilute methylene blue solution, and after a short period found the granules of the stomach, the red blood corpuscles, and other cells stained blue. This method, however, cannot pass as entirely free from error, as Ehrlich frequently found that when the experiment lasts some time, the methylene blue often forms granular precipitates that may be confused with granules. Teichmann directs a detailed analysis to this point, and regards most of the granules described by Schultze as artificial products. Neutral red is highly suitable for the study of vital granule staining, a dye recommended by Ehrlich and employed successfully since that time by Przemysl, Projek, S. Meyer, Solger, Friedmann, Pappenheim, and others. This dye was prepared by O. N. Witt from nitrosodimethylamine and metatoluendiamine, and is the hydrochloric acid salt of a base which is soluble in pure water, yielding a fuchsin red colour, but which in weak alkaline solution, the alkalinity of mineral water suffices, is a yellow-orange hue. Now neutral red is characterised by a really maximal affinity for the majority of the granules. Ehrlich was able by the aid of this dye to demonstrate granules even in some vegetable cells. Moreover, the method of using it is the simplest conceivable, as subcutaneous or intravenous injection, or even feeding, in the higher animals, stains the granules. With frogs' larvae and invertebrates, to allow them to swim in a dilute solution of the dye is often sufficient. The staining also succeeds in surviving organs, and is best effected by allowing small pieces to float in physiological salt solution, to which a trace of neutral red is added, under plentiful access of air. When the object is macroscopically red, it is ready for examination. The finest results are naturally given by organs that are easily teased out, for example flies' eggs, or the Malpighian canals of insects. The staining solution is to be chosen so that the act of staining does not last too long, but on the other hand too high a concentration must not be used. About 1 in 50,000 to 1 in 100,000 is recommended, so that the protoplasm and nucleus remain quite uncoloured. Artificial products with this method cannot entirely be excluded, and, for example, in plant cells containing tannin, are to be explained by the production and precipitation of the salt of tannic acid. However, it is not difficult for the experienced to recognize artificial products as such in individual cases. The kind of granulation, the typical distribution, a comparison with neighbouring cells, the combination of various methods, the comparison of the same object under vital and survival staining, facilitate judgment and obviate mistakes of this kind. The majority of the granules of vertebrates are stained orange-red by neutral red, corresponding with the weakly alkaline reaction of these forms. Granules staining in pure fuchsine colour, and which hence possess a weak acid reaction, are much more rarely found. Combination staining may be recommended as a valuable aid to the neutral red method. Ehrlich has used a double stain with neutral red and methylene blue. Frogs' larvae were allowed to remain in a solution of neutral red, to which a trace of methylene blue had been added. He then found red granulations almost exclusively, only the granules of the smooth musculature of the stomach were stained intensely blue. With the aid of a threefold combination, Ehrlich obtained a still further differentiation of the living cell granules. There is no doubt whatever that a thorough study of this neutral red method would lead to important conclusions as to the nature and function of the granules, and lead us to the most real problems of cell life. With our present information even, we can get definite conceptions founded upon facts, as to the biological importance of the cell granules. In his first publication, Ehrlich described the granules as products of the metabolism of the cells, deposited within the protoplasm in a solid form, in part to serve as reserve material, in part to be cast off from the cell. On the ground of observations on the liver cells, described in detail in a paper of Frerichs, 1883, page 43, Ehrlich gave up this position, though only temporarily. Ehrlich showed that the liver cells of a rabbit's liver, rich in glycogen, appear in dry preparations as bulky polygonous elements, of a uniform homogeneous brown colour, surrounded by a thin, well-defined yellow membrane. In cells that were not too rich in glycogen, small roundish bodies, clearly of a protoplasmic nature, of a pure yellow, can be seen embedded in the homogeneous cells that are coloured brown with glycogen. "The hyaline cellular ground substance carrying the glycogen could not under any circumstances be stained, but the cell granules above mentioned stained easily with all kinds of dyes. It was further possible to show by staining that the membrane was chemically different from the granules, since with eosin aurantia indulin glycerine the membrane stained black, but the granules orange-red." To these observations Ehrlich added the following conclusion, "that the cells of the liver after food really possess a thin protoplasmic membrane and a homogeneous glycogen-bearing substance in which the nucleus and round granules, functionally active, of protoplasm are embedded." On comparing these results with those of more recent investigation of the cells, it is easy to determine the location of the glycogen very accurately. Kupfer has shown, first for the liver cells, and this is now recognized as generally valid, that their contents do not represent a microscopically single substance. In the survival preparation he found, in addition to the nucleus, two clearly distinct substances, a hyaline ground substance in preponderating amount, and a more scanty, finely granular, fibrillary substance embedded in it. Kupfer calls the first paraplasm, the latter protoplasm. On warming the preparation to about 22°C, manifest though feeble movements appeared in the network. It can hardly be doubted that of these two substances the granular reticulated one, the protoplasm, is the more important, and it should not be erroneous to suppose that the granulations of the network form the centre of the particular, specific cell function. In any case it is desirable to give a special name, such as microsomes (Handstein), to these forms, which in the liver cells are recognizable as distinct, round or oval granules, colouring yellow with iodine, and easily and deeply staining in other ways." It was necessary to quote in full from this older paper to show that Ehrlich regarded the granules as the special carriers of the cell function so long ago as 1883, a view that Altmann advocated many years later under the name theory of bioblasts. Altmann's ever-repeated assertion that no one before him had allotted so high an importance to the granules is consequently in disagreement with the facts we have above made sufficiently clear. The importance Altmann ultimately gave to the granules, which he also calls by the name ozonophores, is shown by his own words in Elementary Organisms, 1st edition, page 39. "Our conception of the ozonophores may therefore replace that of the living protoplasm, at least so far as vegetative function is concerned, and may serve us as an explanation of complicated organic processes. Once again shortly summarizing the properties of the ozonophores, as oxygen carriers they can perform reduction and oxidation, and can thus affect the decompositions and syntheses of the body without losing their own individuality. In the meantime Ehrlich had made various observations which could not be completely brought into line with his own earlier hypothesis or the far-reaching conclusions of Altmann. Studies in particular on the oxygen requirements of the organism showed that the ozonophores could certainly not be an important part of the cell. In addition, it was found that normally cells occur in which no granules can be recognized by ordinary methods. Finally, a pathological observation made untenable the view that the granules are the bearers of the cell function. In a case of pernicious anemia, see farbenanalytische Untersuchungen, Ehrlich found the polynuclear cells of the blood and bone marrow and their early forms free from all neutrophil granulation. On the grounds of this observation, Ehrlich returned to his original assumption that the granules are secretory products of the cells, and defined his standpoint at that time as follows: quote, "Did the neutrophil granulations really represent the bodies which supply these cells with oxygen, as Altmann supposes, a condition such as we have here brought forward would be impossible, since with the disappearance of the granules death of the cells must follow. But from the point of view of the secretion theory the condition described is easily explainable. Just as under certain conditions fat cells may completely lose their contents without dying, so the bone marrow cell, if the blood fails to yield to it the necessary substances, may occasionally be unable to produce more neutrophil granules, and thus it becomes non-granular." The view that the granules are special metabolic products of the specific cellular activity is strongly supported by the great chemical differences between them. Ehrlich made these peculiarities clear for the blood cells, and found that their granulations differ from one another, not only in their colour reactions, but also in their shape and solubility, so that they must be sharply distinguished. Whilst for instance the majority of the granules are more or less rounded forms, in some classes of animals, for example in birds, the analogues of the granules of mammalian blood are characterized by a decided crystalline form, and a strong oxophilia. The substance of the mast cell granulations is also crystalline in some species of animals. The size of the individual granules is constant in any animal species for every kind of granule, excepting only the mast cells. The eosinophil granulation reaches its greatest size in the horse, where really gigantic examples are found. The presence of granulated colourless blood cells has been demonstrated in the most various classes of animals, and even in the blood of many invertebrates, particularly, as Knoll has shown, in the lamellibranchiates, polychaetes, pedates, tunicates, and cephalopods. Concerning vertebrates, especially the higher classes, accurate and ample researches are to hand. In birds we recognize two oxophil granulations, of which one is embedded in the cells in the crystalline, the other in the usual granular form. Amongst the vertebrates, most investigated classes possess granulated polynuclear cells. To this circumstance Hirschfeld has recently devoted a thorough paper containing many details worthy of note. In the majority of the animals observed, he found too that the polynuclear cells contained neutrophil granules. In only one animal, the white mouse, did he find them, or granulations analogous to them, completely wanting. According to the investigations carried out some years back in Ehrlich's laboratory by Dr. Franz Müller, these results of Hirschfeld's must be described as inaccurate. After many vain endeavours, Dr. Müller was able to find a method by which numerous, though very minute granules could be found in the polynuclear cells of the mouse. The case shows that it is not permissible to assume the absence of granules when the ordinary staining methods are not at once successful. There is no universal method for the staining of granules, any more than for the staining of various kinds of bacteria. Indeed, all granules that are easily soluble vanish when the triacid method is used, and so a homogeneous cell protoplasm is simulated. But naturally the occurrence of non-granulated polynuclear cells in certain classes of animals is not to be denied from these considerations. Hirschfeld asserts that such cells occur side by side with granulated cells, for instance in the dog, and draws from them far-reaching conclusions as to the meaning of the granules. From Kurloff's work we must insist, on the contrary, that there is no evidence that the non-granulated polynuclears are identical with the granulated cells. Kurloff has shown, at least for guinea pig's blood, that these two heterogeneous elements are to be sharply separated one from the other, and that they have an entirely different origin. Specially important for the theory of the nature of the granules is the circumstance that generally speaking in all species of animals, they are present in those cells of the blood only which are adapted to and capable of emigration. That a certain nutritive function is to be ascribed to the emigration of the granulated cells is a very obvious supposition, scarcely to be denied, and naturally cells with a plentiful store of reserve material are eminently suited for this purpose. The lymphocytes, on the contrary, incapable of emigration, are almost totally devoid of specific granulations. A further indication that the granulations really are connected with a specific cell activity lies in the fact that one cell bears but one specific granulation. The contrary assertions that neutrophil and eosinophil, or eosinophil and mast cell granulations occur in the same cell, Ehrlich regards as unfounded, from extensive researches specially directed to this point. Nor has Ehrlich seen a pseudo-eosinophil cell of the rabbit changed to a true eosinophil. Footnote: the cause of these misunderstandings is the tinctorially different stages of development of the granules, as we have fully explained above. How little adequate tinctorial differences by themselves are to settle the chemical identity of a granulation is at once evident on consideration of the granules of other organs. No one surely would assert that a liver, muscle, or brain cell could occasionally secrete trypsin, simply because the granules of the pancreas stain similarly and analogously to those of the cells mentioned. We would here expressly insist that we only assume a distinct character for each kind of granulation, in the strict sense of the term, for the cells of the blood, since they possess a relatively simple function. In very complex glandular cells, however, with various simultaneous functions, several kinds of granules may be contained. End footnote. That such a transition does not occur is most distinctly shown by the fact that the various granulations behave entirely differently towards solvents. With the aid of acids, for example, the pseudo-eosinophil granules can be completely extracted from the cells, whilst the eosinophil granules remain whole under this process and can now be stained by themselves. The clearest proof that the neutrophil, eosinophil, and mast cells are entirely separated from one another by the fundamental diversity of their protoplasm, of which the granulation is but a specially striking expression, is afforded by the study of the various forms of leukocytosis. As will be shown in detail in the following chapter, neutrophil and eosinophil leukocytes behave quite differently in their susceptibility to chemiotactic stimulation. Substances strongly positively or negatively chemiotactic for one cell group are as a rule indifferent for the other. Frequently indeed there is an exactly opposed relationship, inasmuch as substances which attract the one kind repel the other. Still greater is the difference between the mast cells and the other two cell groups, for so far as present investigations go they are quite uninfluenced by substances chemiotactic for the neutrophil or eosinophil cells. As specific cellular secretions, various kinds of granules must also be sharply marked off from each other by their chemical properties. The granules of the blood corpuscles seem to be of very simple chemical constitution. We have special grounds for the assumption that the crystalline granulations are for the most part composed of a single chemical compound, not necessarily highly complex even, but which seems to be a relatively simple body, such as guanine, fat, melanin, etc. Doubtless other granulations have a more complicated constitution, and very often are a mixture of various chemical substances. The most complicated granules of the blood are the eosinophil, which are, as has elsewhere already been mentioned, of a more complex histological structure. For a peripheral layer is plainly distinguishable from the central part of the granule. It should be mentioned that according to Barker the eosinophil granulations appear to contain iron. The keystone of the hypothesis of the secretory nature of the granules is the direct observation of a secretory process in the cells bearing the granules. Naturally, these researches offer extraordinary difficulties, since only the coincidence of a number of lucky circumstances would allow the passage of dissolved granule substance into the neighbourhood to be followed. Cantak and Hardy have succeeded in demonstrating the secretory nature of the eosinophil granules of the frog. When, for example, anthrax bacilli are introduced into the dorsal lymph sac of the frog, they exert a positive chemiotaxis on the eosinophil cells. The latter come in contact with the bacilli and remain for some time attached to them. During this period Cantak and Hardy observed a discharge of granules from these cells which now possess a protoplasm relatively homogeneous. Afterwards these cells move away from the bacilli and are succeeded by the polynuclear neutrophil cells, as will be mentioned later. These authors were further able to observe gradual accumulation of granules in eosinophil cells in lymph kept under microscopic observation as a hanging drop, and thus demonstrated that they undergo the two stages characteristic of secretion: 1. Appearance of granules within the cells; 2. Discharge of these granules externally. The mast cells, too, seem suited for this purpose, since their specific substance is strongly characterised by its peculiar metachromatic staining, and is further especially readily recognisable, since by its great affinity for basic dyes it remains plainly stained, even in preparations that are almost quite decolorised. In fact, appearances of the mast cells are not infrequently found, which must be referred to excretory processes of this kind. In the first place, it is occasionally seen that the mast cell granulation is dissolved within the cell, and diffuses in solution into the nucleus. In place of the well-known picture of the mast cell, of a colourless nucleus surrounded by a deeply stained metachromatic granulation, a nucleus is present intensely and homogeneously stained in the tint of the mast cell granulation, surrounded by a protoplasm showing but traces of granules. Still more convincing is the presence of a peculiar halo of the mast cells, described by various authors. Ehrlich first shortly mentioned this halo in his book on the oxygen requirements of the organism. A few years ago, Unna, whose notice Ehrlich's remark had no doubt escaped, described an analogous condition as follows: "In some nodules the mast cells appeared in part twice as large as usual, especially with the new mast cell stain, polychrome methylene blue, glycerine ether mixture. This was caused by the staining of a large round halo, in the centre of which lay the peculiar long-known mast cell, consisting of blue nucleus and an areola of deep red granules. Higher magnification showed that the halo was not granular, but very finely reticular, although it exhibited exactly the same red colour as the granules. It was consequently a spongioplasm peculiar to these mast cells." The appearance of the mast cells described by Unna may also be artificially produced by allowing a preparation that is stained with the oxygen-containing analogue of thionin, oxamine, to remain for some time in levulose syrup or watery glycerine. Evidently part of the dyed mast cell substance is dissolved and retained in the immediate neighbourhood. But as Unna possesses great experience of the mast cells, and is a complete master of the methods of their demonstration, one must suppose that the halos described by him were preformed, and did not arise during the preparation of the specimen. It must hence be concluded that an analogous process may go on during life, that these halos are the expression of a vital secretion of the substance of the mast cells externally. From a paper of Calleja we learn that Ramon y Cajal recognized the halos of the mast cells and interpreted them in the manner we have above. Calleja also describes these halos and the method of demonstrating them in detail: thionin staining and mounting the sections in glycerine. We must mention, however, that we do not consider this method suitable for the recognition of preformed halos, for the reasons above mentioned. A condition that Pruss has brought forward in the so-called purpura of the horse is also to be interpreted as a secretory process of the mast cells. He describes young mast cells from the haemorrhagic foci of the wall of the gut, on the margins of which bodies of various sizes appeared, and which differ essentially from the mast cells themselves by their staining. Nevertheless, from their whole configuration and position it is evident that these bodies have arisen in the mast cells themselves, and Pruss comes to the conclusion, "that the degenerating young mast cells secrete a fluid or semi-fluid substance, which as a rule sets on the surface of the cells, but also, more rarely, in their interior." Evidence that the substance of the granules is given off externally may sometimes be seen in the polynuclear neutrophil or their analogues. Thus in rabbit's blood, in which he had experimentally produced leucocytosis, Henkin found a distinct progressive decrease of the pseudo-eosinophil granules on allowing the samples of blood to remain some time in the thermostat. Further, in suppurating foci in man, especially when suppuration has lasted long, or the pus has remained for some time in the place in question (Janowski), a rarefaction, almost a complete disappearance, of the polynuclear neutrophil granules occurs, and is to be explained by a giving up of the granulations to the exterior. These facts and considerations on the whole lead then to the conclusion that in general the granules of the wandering cells are destined for excretion. This elimination of the granules is probably one of the most important functions of the polynuclear leucocytes. End of section 8. Of Histology of the Blood. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by Anna Simon. Histology of the Blood by Paul Ehrlich and Adolf Lazarus. Translated by M. Myers. Chapter 4. Leukocytosis. The problem of leukocytosis is one of the most keenly debated questions of modern medicine. An exhaustive account of the various works devoted to it, of the methods and results, could fill by itself a whole volume, and would widely exceed the limits of an account of the histology of the blood. We can only deal fully, therefore, with the purely hematological side of the subject. Virchow designated by the name leukocytosis a transient increase in the number of the leukocytes in the blood, and taught that it occurred in many physiological and pathological conditions. In the period that followed, particular attention was paid to the leukocytosis in infectious diseases, and to the investigators of the last 15 years in this province we owe very important conclusions as to the biological meaning of this symptom. Above all, Metchnikoff has done pioneer service in this direction by his theory of phagocytes, and though his theory has been shaken in many essential points, yet it has exercised a stimulating and fruitful influence on the whole field of investigation. To sketch Metchnikoff's doctrine in a few strokes is only possible by a paraphrase of the very pregnant words phagocytes, digestive cells. These words express the view that the leukocytes defend the organism against bacteria by imprisoning them by the aid of their pseudopodia, taking them up into their substance, and so depriving them of the power of external influence. The issue of an infectious disease would chiefly depend on whether the number of leukocytes in the blood is sufficient for this purpose. This engaging theory of Metchnikoff has undergone important limitations as the result of further investigation. Denis, Buchner, Martin Hahn, Goldscheider and Jacob, Löwy and Richter, and many others have demonstrated that the most important weapon of the leukocytes is not the mechanical one of their pseudopodia, but their chemical products. Alexine, Bücher. By the aid of bactericidal or antitoxic substances which they secrete, they neutralize the toxines produced by the bacteria, and thus render the foe harmless by destroying his weapon of offence, even if they do not exterminate him. An explanation of the almost constant increase of the leukocytes of the blood in bacterial diseases is given by the chemiotactic as well as by the phagocytic theory of leukocytosis. The principle of chemiotaxis discovered by Pfeffer asserts that bacteria, or rather their metabolic products, are able to attract by chemical stimulus the cells stored up in the blood-forming organs, positive chemiotaxis. In the cases in which a diminution of the leukocytes in the blood is found, it is the result of a repulsion of the leukocytes by the bodies mentioned, negative chemiotaxis. As the experimental investigation of leukocytosis was carried further, it was found that leukocytosis, quite similar to that occurring in infectious diseases, could also be brought about by the injection of various chemical substances (bacterioproteins, albumoses, organic extracts, and so forth), and it became evident that the explanation of the process by chemiotaxis must be supplemented in many respects. Löwit, for instance, found that when substances of this kind are injected, two different stages can be distinguished in the behaviour of the leucocytes. First came a stage in which they were diminished (leukopenia, Löwit), and in such a way that only the polynuclear cells were concerned in the diminution, whilst the number of the lymphocytes was unchanged. After this came the phase of increase of the white blood corpuscles, and here too exclusively of the polynuclear cells, the polynuclear leukocytosis. This behaviour seemed to indicate that during the first period a destruction of white blood corpuscles brought about by the foreign substances took place, and that it was only the dissolved products of the latter which caused the emigration of fresh leucocytes by chemiotaxis. But new objections were raised against this view. Goldscheider and Jacob, in particular, showed by exact experiments that the transient leukopenia of the blood was not true but merely apparent, and was caused by an altered distribution of the white blood corpuscles within the vascular system. For, whilst in the peripheral vessels from which the blood for investigation was usually obtained, there was in fact a diminution of the leucocytes (hypoleukocytosis), in the capillaries of the internal organs, especially of the lungs, a marked increase of leucocytes (hyperleukocytosis) was found. There are other objections to the great importance that Löwit has given to leukopenia. A priori, it is quite incomprehensible that the various substances which in the fundamental test tube experiment are able to exercise a distinct chemiotactic influence on the leucocytes, should under other circumstances need the intervention of the products of decomposition of the white blood corpuscles. Moreover, clinical experience speaks in general against Löwit's theory. For in infectious diseases hyperleukocytosis is very common, and a transient leukopenia is equally rare. This contradiction to the experimental results obtained by Löwit is easily explained when one reflects how different from the natural processes of the disease are the circumstances of experiment. In this case the animal is by intravenous injection flooded at once with the morbid substance, and a violent acute reaction of the vascular and blood systems is the natural consequence. In natural infection, insidious and increasing amounts of poison come quite gradually into play, and for this reason perhaps hypoleukocytosis in the normal course of infectious diseases is much rarer than in the brusque conditions of experiment. Upon the clinical importance of leukocytosis, particularly for the infectious diseases and their various stages, an enormous mass of observations has accumulated. Selecting pneumonia as the best studied example, in the typical course of this disease the constant occurrence of leukocytosis is undisputed. The increase usually continues up to the crisis, and then gives place to a diminution of the leukocytes until a subnormal number is reached. Of special importance are the observations on an absence of leukocytosis in particularly severe or lethally ending cases. Kikotze, Sadler, von Jacques, Tschistowitsch, Türk, and others. In many other diseases as well, the observation has been made that hyperleukocytosis as a rule is only absent in specially severe or in some way atypical cases. Several observers, Löwy and Richter, M. Hahn, Jacob, have been able to demonstrate experimentally for various infections that artificial hyperleukocytosis influences the course of an artificial infection most favorably. The question, in what way does this process contribute to the protection of the body, is at the present time under discussion, and introduces the most difficult problems of biology. The morphological character of leukocytosis is certainly not simple, and we must sharply separate various groups according to the kind of leukocyte increased. The most important consideration is, whether cells capable of spontaneous movement and of active emigration into the blood are increased, active leukocytosis, or whether the number of those cells is raised to which an independent mobility cannot be ascribed, which therefore are only passively washed into the bloodstream by mechanical forces, passive leukocytosis. The passive form of leukocytosis corresponds to the different kinds of lymphemia, including that of leukemia. In the section on the lymphatic glands we have established this view in detail, and we have particularly insisted that a suppuration consisting of lymph cells does not occur. In sharp contrast to this form there are for every specific kind of active leukocytosis analogous products of inflammation, pus, exudations, composed of the same kind of cell. We divide active leukocytosis into the following groups: A. Polynuclear leukocytosis. 1. Polynuclear neutrophil leukocytosis. 2. Polynuclear eosinophil leukocytosis. B. Mixed leukocytosis in which the granulated mononuclear elements take part. Myelemia. A1. Polynuclear neutrophil leukocytosis is the most frequent of all forms of active leukocytoses. Virchow, the discoverer of leukocytosis, advocated the view that it resulted from an increased stimulation of the lymph glands. The stimulation of the lymph glands consists in that they are engaged in an increased formation of cells, that their follicles enlarge, and after a time contain many more cells than before. The swelling of the lymphatic glands has as a consequence an increase of the lymph corpuscles in the lymph, and through this an increase again of the colourless blood corpuscles. This standpoint had to be abandoned when Ehrlich showed that it is chiefly the emigration of the polynuclear neutrophil cells which brings about leukocytosis. Exact figures on this point were first given by Einhorn, who worked under Ehrlich, and were later generally confirmed. Corresponding with the increase of neutrophil blood corpuscles alone, there is always a relative decrease of lymphocytes, often to 2% and even lower. It must here be borne in mind that the percentage of the lymph cells may be much diminished without change in their absolute number. It has however been conclusively demonstrated that occasionally in polynuclear leukocytosis the absolute number of the lymphocytes may decrease. Einhorn had already described a case of this kind, and recently Türk has for the first time established the fact by an abundance of numerical estimations. Naturally, an ordinary leukocytosis may be combined with a lymphemia. We have already mentioned elsewhere that in the leukocytosis of digestion or of diseases of the intestine in children such a coincidence occurs. The eosinophil cells are as a rule diminished in ordinary polynuclear leukocytosis, as Ehrlich had already mentioned in his first communication. The diminution is often considerable, often indeed absolute. A few diseases show, besides the neutrophil leukocytosis, an increase of the eosinophils as well, as we shall describe in detail in the next section. Polynuclear neutrophil leukocytosis, leukocytosis catexogen, may be divided into several groups according to their clinical occurrence. We distinguish A. Physiological leukocytosis, which appears in health as an expression of changes in the physiological state. To this group belongs the leukocytosis of digestion, the leukocytosis from bodily exertion, Schönberg and Zuntz, or from cold baths, and further the leukocytosis of pregnancy. B. Pathological leukocytosis. B1. The increase of polynuclear cells occurring in infectious processes, often called inflammatory, after the principle a potiori fit denominatio. The majority of febrile infectious diseases, pneumonia, erysipelas, diphtheria, septic conditions of the most varied etiology, parotitis, acute articular rheumatism, etc., are accompanied by a leukocytosis of greater or less extent. In this connection uncomplicated typhoid fever and measles occupy a peculiar position. In them the absolute number of white blood corpuscles is diminished, and chiefly at the expense of the polynuclear neutrophil cells. For the details we have quoted, and for the course and variations of leukocytosis in infectious diseases, we refer to the thorough monograph of Türk. Of Türk's observations we will mention only that in the final stage of the process of leukocytosis, which occurs at the time of the crisis in diseases which run their course critically, mononuclear neutrophil cells and stimulation forms as well often make their appearance in the blood. In still later stages, in which the blood has once more a nearly normal composition, a moderate increase of the eosinophils, gradually waxing and again waning, is very frequently found. Zappert and others. Stiénon, who has likewise devoted special researches to the occurrence of leukocytosis in infectious diseases, shows this point very well in his curves. B2. Toxic leukocytosis, occurring in intoxications with the so-called blood poisons. This important group has not yet received adequate treatment in the literature. In general, the majority of blood poisons, potassium chlorate, the derivatives of phenylhydrazine, pyridine, phenacetin, call forth even in men a considerable increase of the leukocytes besides the destruction of the red blood corpuscles. This has been observed experimentally by Rieder. We observed marked increase of the white blood corpuscles after poisoning from arseniuretted hydrogen, from potassium chlorate, further in a fatally ending case of hemoglobinuria, sulfonal poisoning, as well as after protracted chloroform narcosis. B3. The leukocytosis which accompanies acute and chronic anemic conditions, especially post-hemorrhagic. B4. Cachectic leukocytosis in malignant tumors, phthisis, etc. Footnote. The so-called agony leukocytosis we do not regard as a true leukocytosis, but only as the expression of a stoppage of the circulation caused by that condition. This produces an accumulation of the white corpuscles on the vessel walls, especially in the peripheral parts of the body which are as a rule used for clinical investigation. A leukocytosis is thus simulated. End footnote. To enter here more precisely into the special clinical importance of blood investigation in different forms of disease would lead us too far, and we refer for this subject to the excellent and thorough monograph on leukocytosis by Rieder and to the papers of Zappert and Türk. In this place we will only touch on the most weighty points. A. The importance for differential diagnosis of the leukopenic blood condition in typhoid fever as compared with other infectious diseases, and in measles as against scarlet fever. B. The prognostic importance of the enumeration of the white blood corpuscles. Thus, for example, the absence of leukocytosis influences the prognosis of pneumonia unfavorably. Kikotze and others. And the appearance of numerous myelocytes in diphtheria is ominous, as demonstrated by C.S. Engel. Finally, we may dismiss in a few words the origin of polynuclear neutrophil leukocytosis, and refer to what has been said in another place on the function of the bone marrow. In agreement with Kurloff's researches, Ehrlich formulated (On Severe Anemic Conditions, 1892) his views on this subject as follows. Quote, "The bone marrow is a breeding place in which polynuclear cells are produced in large numbers from mononuclear pre-existing forms. These polynuclear cells possess above all other elements the power of emigration. So soon as chemiotactic substances circulate in the blood, which attract the white elements, this power comes into play. This readily explains the rapid and sudden appearance of large numbers of leukocytes, which so many substances bring about, and particularly the bacteria proteins, recognized by Buchner as leukocytic stimuli. I regard leukocytosis, therefore, in agreement with Kurloff, as a function of the bone marrow." End quote. Of great theoretical interest is the contrast between eosinophil and neutrophil cells. At the height of ordinary leukocytosis, the number of eosinophil cells is diminished often to disappearance, whilst during its decline they occur in abnormally high numbers. Hence it follows that the eosinophil and neutrophil cells must react towards stimulating substances completely differently and, in a certain sense, oppositely. Footnote. It is also of interest to notice the behaviour of the eosinophil cells in the passive form of leukocytosis, lymphemia. A priori, both conditions could be combined. As C.S. Engel has established, in the congenital syphilis of children, a simultaneous marked increase of lymphocytes and eosinophil cells is found. The lymphocytosis in these cases is probably due to the anatomical changes of the lymph glands, and the eosinophilia to specific chemiotactic attraction. End footnote. It seems, generally speaking, that the bacterial metabolic products formed in the human diseases which are positively chemiotactic for the polynuclear neutrophil cells are negatively chemiotactic for the eosinophils, and vice versa. The explanation of the individual clinical forms of leukocytosis is self-evident from the above description. The occurrence of physiological and inflammatory leukocytosis is exclusively to be explained by chemiotaxis. In the other forms, however, other factors also come into play, in particular the increased activity of the bone marrow, or the extensive transformation of fatty to red marrow, causing a large fresh formation of leukocytes. Alpha 2. Polynuclear eosinophil leukocytosis, mast cells. Our knowledge of eosinophil leukocytosis is still of comparatively recent date. After Ehrlich demonstrated the constant increase of the eosinophil cells in leukemia, a considerable time elapsed before an eosinophilia was found in other diseases, an eosinophilia, however, that differs in its essential traits from the leukemic type. To Friedrich Müller we owe the first researches in this direction, at whose suggestion Gollasch investigated the blood of persons suffering from asthma, in which he was able to demonstrate a considerable increase of the eosinophil cells. This was followed by the researches of H. F. Müller and Rieder, who discovered the frequency of eosinophilia in children, and its presence in chronic splenic tumours. Further, by the well-known work of Ed Neusser, who observed a quite astounding increase of the oxyphil elements in pemphigus, and by the almost simultaneous analogous observations of Cannon in chronic skin diseases. From amongst the flood of further papers upon this condition we will only mention the comprehensive account of the subject by Zappert. By eosinophilia we understand an increase only of the polynuclear eosinophil cells in the blood. Confusion of this form of leukocytosis with leukemia is quite impossible, because a good number of characteristic signs are necessary for the diagnosis of the latter, as we shall have to explain in the next section. The presence of mononuclear eosinophil cells in the blood should not be regarded, as is the case in many quarters, as an absolute proof of leukemia, for they are also found in isolated cases of ordinary leukocytosis. The increase of eosinophil cells is not always relative, but may be absolute. The relative number, normally 2 to 4% of all leukocytes, rises in eosinophilia to 10, 20, 30% and over. In a case described by Grawitz, 90% indeed was found. The thorough researches of Zappert, carried out on moist preparations by a suitable method, are particularly instructive with regard to their absolute number. As the lowest normal value he gives 50 to 100 eosinophil cells per cubic millimeter, as mean value 100 to 200, as a high normal value 200 to 250. The highest absolute number he has ever found was 29,000 per cubic millimeters in leukemia. The highest number in simple eosinophil leukocytosis 4,800 in a case of pemphigus. Reinbach, indeed, once found about 60,000 eosinophil cells per cubic millimeters in a case of lymphosarcoma of the neck with metastasis in the bone marrow. Polynuclear eosinophil leukocytosis, apart from the form observed in healthy children, occurs in varied conditions, and for comprehensiveness we divide them into several groups. We distinguish eosinophilia 1. In bronchial asthma. Increase of the eosinophil cells of the blood, often considerable, amounting to 10 and 20% and more, has been regularly found, first by Gollasch, later by many other observers. For the special clinical course of the eosinophilia in asthma, see below. 2. In pemphigus. Neusser first recorded that an extraordinarily great, indeed a specific eosinophilia was found in many cases of pemphigus. This interesting observation has been confirmed on many sides, in particular by Zappert, who once observed 4,800 oxyphil per cubic millimeters. 3. In acute and chronic skin diseases. Cannon was the first to notice that in a fairly large number of skin diseases, especially in prurigo and psoriasis, the eosinophil cells are increased up to 17%. The observation of Cannon is worthy of attention, that the increase of the eosinophils is connected with the degree of extension of the disease, rather than with its nature or local intensity. In a case of acute widely distributed urticaria, A. Lazarus found the eosinophils increased to 60% of the leukocytes, a number which after the course of a few days again sank to normal. 4. In helminthiasis. The first observations on the occurrence of eosinophilia in helminthiasis we owe to Müller and Rieder, who obtained fairly high values (8.2 and 9.7%) in 2 men suffering from ankylostomum duodenale. Shortly afterwards Zappert stated that he had found a considerable increase of the eosinophil cells in the blood, reaching 17% in 2 cases of the same disease. At the same time he demonstrated Charcot's crystals in the faeces. In a 3rd case of ankylostomiasis, Zappert found no increase of eosinophil cells in the blood, nor the crystals in the faeces. Almost simultaneously, Siege made similar observations. For a detailed working out of this important branch we are greatly indebted to Leichtenstern. Under his direction Buchler's established the interesting fact that ankylostomiasis in its relation to eosinophilia does not occupy a special place in diseases caused by worms. All kinds of helminthides, from the harmless oxyuris to the pernicious ankylostoma, may bring about an increase of the eosinophil cells in the blood, often to an enormous extent. In his monograph on Bothriocephalus anemia, Schaumann, with reference to the behaviour of the eosinophil cells, states that he has found them in but few cases of this disease. Buchler's reports an observation of 16% eosinophils in oxyuriates, of 19% in ascarides, and Professor Leichtenstern, as we learned from a private communication, has quite recently found 72% eosinophil cells in a case of ankylostomiasis, and 34% in a case of tinea media canalata. It is well worthy of note that Leichtenstern was able to observe numerous eosinophil cells in the blood in those cases where Charcot's crystals were abundantly contained in the faeces. Since eosinophil cells and Charcot's crystals have elsewhere been observed to be interconnected phenomena, for example in bronchial asthma, in nasal polypi, in myelemic blood and bone marrow, one must fall in with Leichtenstern's supposition that eosinophil cells ought also to be found in the intestinal mucus in cases of ankylostomiasis. Positive observations on this point as yet are wanting. T. R. Brown, who worked under direction of Thayer, has lately communicated the interesting observation that in trichinosis there is constantly an extraordinary relative increase in the oxyphil leukocytes in the blood up to 68%. The absolute figures were also much raised, and attained values, 20,400 for example, which are by no means frequent even in leukemia. Brown regards this astonishing phenomenon as pathognomic for trichinosis, so much so that in a case that was clinically obscure he made from the marked eosinophilia the diagnosis of trichinosis, which was later fully confirmed. 5. Post-febrile form of eosinophilia, after the termination of various infectious diseases. In the section on polynuclear neutrophil leukocytosis we have already mentioned that at the height of most of the acute infectious diseases, with the single exception of scarlet fever, the eosinophils undergo a relative decrease and may even entirely disappear. In the post-febrile period, however, abnormally high values for the eosinophil cells are often found, or even a well-marked eosinophil leukocytosis, which generally attains but moderate degree. Türk, for example, in pneumonia found a post-critical eosinophilia of 5.67%, 430 absolute; after acute articular rheumatism 9.37%, 970 absolute. Zappert, in malaria 1 day after the last attack, 20.34%, 1486 per cubic millimeters. The eosinophilia observed as the result of tuberculin injections we include, in agreement with Zappert, in the group of post-febrile leukocytosis, for it appears only after considerable rises of temperature. During the real reaction period the number of eosinophil cells sinks and only goes up again after the termination of the fever. The rise may be very considerable. In one case of Zappert's the number of oxyphils increased to 26.9%. In another of his cases the highest absolute figure formed after tuberculin injections was 3,220 per cubic millimeters. In a case of Gravitz the eosinophilia was quite extraordinary. The most marked changes in the blood occurred some 3 weeks after cessation of the tuberculin injections, of which 8 altogether (from 5 mg to 38 mg) were given. Investigation showed 4,000,000 red blood corpuscles per cubic millimeters, 45,000 white. Amongst the latter there were 10 eosinophils to 1 non-eosinophil. The total number of eosinophil cells amounted to some 41,000 per cubic millimeters, whilst the other cells as a whole made up some 4,000. Inasmuch as the latter contained polynuclears, lymphocytes, and other forms, it follows that in this case the polynuclear neutrophils must have been very much decreased, not only relatively but also absolutely, so that this case represents precisely the contrary condition to ordinary leukocytosis and the infectious form in particular. 6. In malignant tumours. In the cachexia from tumours an increase of the eosinophil cells has been observed by various authors. It is however of moderate degree and does not exceed 7 to 10%. Out of 40 decided cases, Reinbach found the eosinophils increased only in 4. In a case of sarcoma of the forearm he found 7.8%, of the thigh 8.4%. Malignant tumour of the abdomen 11.6%. Besides these, he describes the case of lymphosarcoma of the neck with metastasis in the bone marrow, in which an unexampled increase of the white blood corpuscles, and especially of the eosinophil cells, was found. The absolute number of the latter amounted on one day to some 60,000. This is an increase of 300-fold the normal, which apart from leukemia has doubtless never before been found. 7. Compensatory eosinophilia, after exclusion of the spleen. We have entered in detail into this form in the chapter on splenic function, and have there already mentioned that the increase of the eosinophils found in chronic splenic tumours by Rieder, Weiss, and others must also be referred to the exclusion of the splenic function. 8. Medicinal eosinophilia. Under this group occurs only a single observation of van Noorden's, who observed the appearance of an eosinophilia up to 9% in 2 chlorotic girls after internal administration of camphor. In other patients this occurrence did not repeat itself, but probably researches specially directed to this province of pharmacology would bring to our knowledge many interesting facts. On the origin of polynuclear eosinophil leukocytosis, authors have put forward various theories, which we will here critically discuss in succession. An experiment frequently quoted as explanatory is that of Müller and Rieder's. These authors do not derive the eosinophil cells of the blood from the bone marrow, but assume, as very probable, that the finely granular cells grow into eosinophils within the blood stream. This developmental process seems very improbable for many reasons. Since the polynuclear cells circulating in the blood are all under the same conditions of nutrition, it is a priori inconceivable why only a relatively small portion of them should undergo the transformation in question. And it is quite inexplicable why in infectious leukocytosis, where the number of the polynuclears is increased so enormously, their ripening to the eosinophils should remain completely interrupted. But the fact that a transition from neutrophil to oxyphil cells has never really been observed in the blood is decisive evidence against the hypothesis of Müller and Rieder. Were the hypothesis true, transitional stages ought to be found with ease in every sample of normal blood. Rieder and Müller themselves are unable to bring forward any positive result of this kind, else they would hardly have been contented to fall back on the authority of Max Schultze, who professed to show the transitional forms between the finely and coarsely granular leukocytes in the circulating blood. The authority of Max Schultze in morphological questions stands high, and very rightly, but one ought not to rely upon it for support in problems that are really histochemical and which should be solved by their appropriate methods. As a logical consequence of their view, and in decided opposition to Ehrlich, Müller and Rieder assume that the eosinophil cells of the bone marrow "are far rather the expression of a storage than of a fresh formation there. The bone marrow, therefore, should be regarded in reference to the coarsely granular cells of the blood more as a storage depot, where these cells serve other purposes, which for the present cannot be more closely defined." The chief reason for this assumption these authors see in the fact that the majority of the eosinophils in the bone marrow are mononuclear, whilst those of normal blood possess a polymorphous nucleus. Müller and Rieder should themselves have raised the obvious objection that the same holds good for the nucleus of the neutrophils. They would then have seen the fault in their theory, for according to it the most important blood preparing organ constitutes, as it were, not the cradle of the blood cells, but their grave. The simplest and readiest explanation, based too upon histological observation, is surely this: that the mononuclear eosinophil cells grow into polynuclear in the bone marrow, but that the latter only reach the blood by means of their power of emigration. As this view has been accepted by the great majority of authors since Ehrlich's paper on severe anemic conditions, we believe we may content ourselves with the above objections to the Müller-Rieder theory, although it has even quite recently found supporters, for example B. Lenhartz. H. F. Müller moreover in his paper on bronchial asthma, 1893, takes a position different from his earlier and approaching that of Ehrlich. In considering the production of polynuclear eosinophilia, we may best start from an experiment of E. Neusser's. Neusser found in a pemphigus patient whose blood showed a considerable increase of the eosinophils, that the contents of the pemphigus bulla consisted almost entirely of eosinophil cells. Neusser now produced a non-specific inflammatory bulla in the skin by a vesicant, and found that the cellular elements in it were exclusively the polynuclear neutrophil concerned in all ordinary inflammations. Exactly analogous conditions, occurring spontaneously, have been demonstrated by Lerède and Perrin in the so-called During's disease. The bullae which appear in this dermatosis contain, so long as their contents are clear, chiefly polynuclear eosinophil cells. In a later stage, as is usually the case, bacteria effect an entrance into the bullae, which now become filled with neutrophils. According to modern views on suppuration, the experiment of Neusser and the observation of Lerède and Perrin can only be explained by the hypothesis that the eosinophil and neutrophil cells, as we have already several times mentioned, are of different chemiotactic irritability. Hence the eosinophil cells only emigrate to those parts where a specific stimulating substance is present. From this point of view, experiments and clinical observations known up to the present on eosinophilia may be readily explained. Neusser's experiment, for instance, may be explained in the following way. In the pemphigus bullae, a substance is present that chemiotactically attracts the eosinophils. Hence the cells normally contained in the blood emigrate into them, and produce the picture of an eosinophilous suppuration. Should the disease assume from the first a localized distribution only, the essential feature of the process is excluded. A totally different appearance, however, is produced when the disease has attacked large areas. Under these circumstances, large amounts of the specific active agent reach the bloodstream by absorption and diffusion. Here it exercises a strong chemiotactic influence on the physiological storage depot of the eosinophils, the bone marrow, leading to an increase of the eosinophils of the blood to a greater or less degree. The bone marrow, according to general biological laws, is by the decreased emigration now further stimulated to a fresh production, and during a protracted illness can hence keep up the eosinophilia. In this way other clinical observations may be explained. Golasch has found that the sputum of asthmatic patients contains, in addition to Charcot-Leydens crystals, eosinophil cells only. One must therefore assume that within the bronchial tree there exists material which attracts the eosinophils. This supposition is also supported by the close connection that obtains, according to many observations, between the severity of the disease and the eosinophilia. Thus van Noorden records that the eosinophil cells are more numerous about the time of an attack. They accumulated in especially large numbers after attacks had rapidly occurred several days in succession. That the increase of the eosinophil cells in this instance is directly connected with the attacks, and is not the expression of a permanent constitutional anomaly, is shown by a case in which van Noorden found 25% eosinophils during the attack, and a few days later could only observe one example in 12 cover slip preparations, a diminution therefore of this group of cells. The observations of Cannon in skin diseases are quite similar, for he showed that the expression of the disease determines the degree of eosinophilia more than its intensity. and it is the former factor which directly determines the quantities of the specific agent that pass into the blood. To the Müller-Rieder hypothesis and the chemiotactic theory of eosinophil leukocytosis, a third has lately been added, which may be shortly called the hypothesis of the local origin of the eosinophil cells. A. Schmidt has, with special reference to asthma, raised the question, "whether in the extensive production of eosinophil cells in asthma, local production in the air passages is not more probable than origin from the blood. One may well regard the increase of the eosinophil cells in the blood of an asthmatic as secondary." This view, which has also been advocated by other authors, rests more particularly on the following facts and considerations. 1. That in various diseases of the nose, especially in mucous polypi and hyperplasia of the mucous membrane (Leyden, Benolivi and others), a great accumulation of eosinophil cells is found in these tissues, whilst they are apparently not increased in the blood. This objection is easily laid aside from the chemiotactic point of view. For if in the places in question substances are present which act chemiotactically on the eosinophil leukocytes, in the course of time marked accumulation must occur, without an increase of their number in the blood. One might as well conclude from Neumann's experiment in lymphatic leukemia, for example, where the artificial suppuration consisted only of polynuclear neutrophil cells, that the polynuclear cells were formed in the tissue, since in the blood they were present in very small percentage. For in this case too the same incongruity between the blood and the particular tissue exists. 2. Adolf Schmidt has urged the converse argument. He showed that in the sputum of patients with myelogenic leukemia no more eosinophil cells were present than are commonly to be found in the bronchial secretion, although the blood was unusually rich in eosinophil cells. In our opinion, however, this observation does not support the hypothesis of local origin, but on the contrary is clear evidence that not the larger or smaller number of eosinophil cells in the blood decides their emigration, but the presence of specifically active chemical stimuli. For we know from our observations on leukocytosis in infectious diseases that the bacterial stimulating substances act on the eosinophil cells rather in a negative than in a positive sense. And if ordinary sputum is not rich in eosinophils, in spite of a marked eosinophilia of the blood, this only corresponds to our experience in general. Indeed, this phenomenon is quite similar to Neusser's pemphigus experiment, where the specific foci of disease showed an eosinophilia, whilst abscesses produced artificially, on the contrary, only neutrophil cells. Finally, we may employ to support our view another analogous experiment of Schmidt himself. He found numerous eosinophil cells in the sputum of an asthmatic patient, but only neutrophil cells in an artificially produced suppuration of the skin. Thus we see that the chief reasons brought forward by the supporters of the theory of local origin are not proof against the most obvious objections that can be raised from the chemiotactic standpoint. Moreover, neither histological nor experimental proof has been given for this theory, in spite of numerous investigations in this direction. All the same, it should not be out of place to explain the possibilities that are given for a local origin of the eosinophil cells. First, the eosinophil cells might be the result of a progressive metamorphosis of the normal tissue cells. That such a process is possible is proved by the local origin of the mast cells. These may arise, as Ehrlich and his school have always assumed, by transformation of pre-existing connective tissue cells. Footnote. This view has lately received striking confirmation from the interesting experiment of Boehmer, who produced on himself, by means of continued stimulation with Urticaria aureus, a considerable increase in 4 days of the mast cells in the irritated portions of the skin. End footnote. But that the same holds good for the eosinophil cells as well has nowise as yet been proved. Secondly, it is conceivable that isolated eosinophil cells, pre-existing in the tissues, should rapidly multiply, and so produce a local accumulation only. Numerous mitoses could be considered an adequate proof of this process. But so far no figures of nuclear division have been observed. Indeed, A. Schmidt, who has directed special experiments thereto from the standpoint of his theory, has found them entirely absent. As a third possibility for the local origin of the eosinophil cells, their direct descent from neutrophil cells is conceivable, and is by many regarded as a kind of ripening. This assumption nevertheless must be described as unsound, since the necessary condition of its foundation, namely the observation of corresponding transitional stages, has not so far been fulfilled. By the inductive method, then, we conclude that a local origin of the eosinophil cells can hardly come under discussion. And this conclusion is strengthened by comparison with the behaviour of the mast cells, which are related to the eosinophils in many points, and only differ from them essentially in the nature of their granulation. The mast cells too, like the eosinophils, form a normal constituent of the bone marrow, and occur regularly besides in normal blood, though in very small number. According to Cannon they amount to 0.28% of the leukocytes. We know that the mast cells are produced in large quantities locally, wherever an overnutrition of the connective tissue occurs, for instance in chronic diseases of the skin, elephantiasis, brown induration of the lungs. In the case of the mast cells, then, we see the conditions actually realized which the supporters of the theory of the local origin of the eosinophil cells only assume. We should therefore expect that an increase of mast cells in the blood or in certain inflammatory exudations would be by no means seldom. With this point in mind, Ehrlich has subjected the sputum in emphysema and brown induration of the lungs to exact examination for 20 years. Nevertheless, he has obtained entirely negative results. The special blood investigations of Cannon have likewise proved to be practically negative. In 22 healthy persons, Cannon entirely failed to find the mast cells on 9 occasions. In the others he found on the average 0.47%. The highest percentage number obtained was 0.89%. Only in a few cases of skin disease was a slight increase indicated. The average amounted to 0.58%, a number therefore which is often to be found in healthy individuals. A leukocytosis of mast cells comparable with the eosinophil or neutrophil forms of leukocytosis has not been demonstrated in the cases of Cannon or other observers. On the other hand, the mast cells undergo a considerable increase in myelogenic leukemia, in many cases equalling or even exceeding that of the eosinophils. We shall not err in deriving the mast cells of the blood solely from the bone marrow, on the grounds of this fact, or in conjecturing that their origin is not from the connective tissue, even when they are excessively increased. That a well-marked basophil leukocytosis has not so far been observed may be thus explained. The substances which attract the mast cells are very rarely produced in the body, much more seldom than the corresponding substances attractive for the eosinophils. In morbid conditions, where substances attracting the mast cells were present, it might be possible to find a separation of mast cells, or a mast cell leukocytosis as well. In this connection an observation of Albert Neisser is of the greatest interest. He met with, private communication, one out of a numberless cases of gonorrhea in which the purulent secretion consisted entirely of mast cells. We think we have shown in the preceding paragraphs that the evidence so far brought forward for a local origin of the eosinophil cells does not withstand the objections that have been raised. The task now lies before us to produce positive proof that the accumulations of eosinophil cells in the organs and secretions must be explained by emigration from the blood. This proof offers great difficulties inasmuch as we normally find eosinophil cells in many places. Here then we cannot trace a process step by step, but we have to deal with final conditions. Could we observe the genesis of eosinophil cells in organs usually free from them, it would be easier to clear up this question. Up to the present but a single observation on this point is available. Michaelis established the interesting fact that on interrupting lactation in suckling guinea pigs, in the course of a few days numerous eosinophil cells collect in the mammary glands, but not in the lumen of the canaliculi. The eosinophil cells are further polynuclear, exactly corresponding to those of the blood, and therefore to be regarded as immigrants. We may explain this condition according to modern views as follows. Under certain conditions the mammary gland is capable of an internal secretion, by means of which substances are produced that are specifically chemiotactic for the eosinophil cells. When the external secretion of milk is disturbed, the internal secretion is abnormally increased. The fact, too, that in Michaelis researches, no eosinophil cells passed into the true secretion of the gland may be thus explained. Footnote: Unger has recently published completely analogous observations on the human breast for the mast cells. Under the influence of stagnation of the milk, he saw an invasion of the gland tissue by typical mast cells. End footnote. Exactly similar observations have been made on pathological material first recorded in the brilliant and fundamental work of Goldmann. In a case of malignant lymphoma, Goldmann found a considerable accumulation of eosinophil cells within the tumour, and demonstrated anatomically that it was brought about by an emigration of the cells from the vascular system. Hence Goldmann concluded that the eosinophil cells pass over into the tissue in question at the call of certain chemiotactic products. Goldmann, and later Kauter, showed that these eosinophil cells were not merely due to an ordinary inflammation, for in a large number of other diseases of the lymph glands, particularly the tuberculous, they were entirely absent. Similarly, Lered and Perrin have shown in their investigations of During's disease that the eosinophil cells which are also present in the cutaneous tissue in large numbers, apart from the contents of the bullae, are due to an emigration from the blood stream. Thus it is evident from a number of various facts that the eosinophil cells found in the tissues are not formed there, but have immigrated from the blood stream. It naturally often happens that this appearance is not preserved equally distinctly in all cases. For, as has been seen in the ordinary polynuclear leukocytes, the immigrated polynuclear eosinophils may similarly change to mononuclear cells. They may perhaps settle down and approximate to the character of fixed connective tissue cells. Such appearances may readily give rise to the view that in this case the reverse nuclear metamorphosis has occurred, that is, a progressive development from mononuclear eosinophil to polynuclear cells. In agreement with Goldmann, Jarason and H.F. Müller, we believe that the only admissible explanation of the facts mentioned above is that the eosinophil cells obey specific chemiotactic stimuli. By this hypothesis we can easily understand eosinophil leukocytosis, the presence of eosinophil cells in exudations and secretions, and the local accumulation of this kind of cell. As to the nature of these chemiotactically active substances, we can so far only surmise. From amongst the clinical phenomena capable of throwing light on this subject, we mention once more the fact that the metabolic products of bacteria repel the eosinophil cells. The opposed behaviour of eosinophil and neutrophil cells is very well illustrated by a case of Liechtenstern. In a very anemic, almost moribund patient with ankylostomias, there were found 72% eosinophil cells in the blood in 1897. The patient contracted a Krupus pneumonia, and in the high febrile period of the disease the number of eosinophils sank to 6 to 7%, and rose again after the termination of the pneumonia to 54%. After removal of the worm the number at once fell to 11%. In the year 1898 the patient harboured but a very few ankylostomata. Charcot's crystals were no longer present in the faeces. The number of the eosinophils amounted to 8%. The question, what cells produce on their destruction actively chemiotactic substances, is of very great importance, but cannot be answered with the material at present available. The breaking up of ordinary pus cells or lymphocytes does not appear to give rise to any such substances, but there is much evidence that the decomposition products of epithelial and epithelioid cells act chemiotactically. Thus we can explain the frequent occurrence of eosinophilia in all kinds of skin diseases. Again, in all atrophic conditions of the gastric, intestinal, and bronchial mucous membrane there occurs a local accumulation of eosinophil cells. Further, this kind of cell is increased in the neighbourhood of carcinoma. Additional support for this view is seen in the fact that in bronchitis and asthma the less the suppurative element of the secretion is developed, the more numerous are the eosinophil cells. An observation of Jarason is worthy of mention in this connection. He observed abundant eosinophil cells in foci of lupus after injection of tuberculin. In these foci then, by the destruction of the epithelioid cells brought about by the tuberculin, substances must have been produced which act chemiotactically on the eosinophil cells. The specific substances are absorbed and reach the blood and impart to it also the chemiotactic power. The direct cause then of most forms of eosinophilia seems actually to lie in a destruction of tissue and in the products thus produced. On the other hand, it cannot be doubted that substances foreign to the organism, circulating in the body, may act chemotactically on the eosinophil cells. A very interesting observation of Goldmann's deserves mention here. Goldmann found in preparations of the pancreas of Proteus sanguinis containing parasites that the eosinophil cells in the neighbourhood of the encapsulated parasites were much increased, whereas they were sought for in vain in more distant parts. The observations quoted above of the well-marked eosinophilia in the different forms of helminthiasis may here be specifically mentioned. The action of the helminthides was formerly regarded as purely local, but the indications that they act also by the production of poisonous substances continue to increase. Thus Linstow has pointed out that the general typhoid state, and the fatty degeneration of liver and kidneys, that is, of organs which the trichina does not reach, necessitate the assumption of a poisonous substance. And in several varieties of ankylostoma as well there is distinct evidence of the production of a poison. We gather from Husemann's article on animal poisons (Eulenberg's Realen Encyclopädie, 1867) that just as ankylostomum in man produces the well-known severe anemia, so ankylostomum trigonocephalum in the dog, and ankylostomum perniciosum in the tiger causes analogous general effects. Bothriocephalus latus too is now generally accredited with the production of a definite toxic substance, and the common tapeworm even by no means infrequently brings about injuries to the body which are to be referred to the action of a poison. So much follows from these observations that the tapeworms cannot only absorb but also can give out substances that are absorbed from the intestine of the host and are able to bring about distant effects. One expression of these distant actions is, as Leichtenstern insists, the eosinophilia of the blood. We do not think we should assume on the evidence before us that the substance which attracts the eosinophil cells is identical with the cause of the anemia. Many observations, the absence, for example, of eosinophilia in Bothriocephalus anemia (Schaumann), render probable the existence of two different functions. In any case, the substance causing the eosinophilia is more widely distributed than that to which the anemic condition is due. Section 9.